I am indeed, but I understood that Marco will talk first, and I actually prepare a couple of comments on what I suppose Marco will comment according to a recent discussion. So if Marco doesn't have any trouble,
yeah,
no, and no problem.
What happens is that we originally published in the order and so on, but it's fine. Yeah. Marco, are you okay?
Yeah, no problem. Yes,
great. Then we will continue in the following set of two talks with Marco Laurati's talk. I think that I missed your title, so go ahead and
yeah, don't worry. Yes.
Because I have the title of your talk, like the title of the session, and anyway, go ahead and explain yourself, Marco. Thank you very much.
Yes. So can you see my screen?
Yeah.
Okay, perfect. Okay, so, well, yes, so today actually, as the title is saying, I would like to essentially start with illustrating how you can combine basically different experimental approaches and especially techniques, let's say, to investigate the properties of glassy systems, and I will illustrate that by using some examples taken from our investigations of colloidal mixtures. And actually, after that, I would like to move to let's say some recent results, which actually are also on mixtures, but different mixtures of soft colloids and polymers. Essentially, these are very recent results, and I'm presenting those results indeed with the aim of having your opinions on what we are observing, because we are at the moment trying to understand what is going on in this system. So let me start anyway with the first part, in which, as I said, I will show you some exemplary results on binary colloidal mixtures, essentially concentrated mixtures of hard spheres. So why we were interested in studying this system? Well, this is first of all because these mixtures of hard spheres are the simplest multi-component colloidal system that can be used as a model for more complex multi-component systems that, for example, you might be interested in studying in applications. And in this sense, for example, it's interesting to study the effects of mixing, because mixing, for example, can be used to increase the ability to pack more material in the system. And also, it's interesting because mixing can induce transitions in the states of this system, which can have, again, implications for applications, for example, for transport of highly packed systems. And obviously, it's also very interesting for the fundamental investigation of glass transition, because as I'm showing this from these results from a paper from Thomas Voigtman, these binary mixtures have a very rich scenario of presenting very different glass states, which are characterized by different gauging mechanisms and different dynamical behaviors of the components. So as I said, what I would like to actually stress today is, first of all, that you need to employ actually techniques, both, for example, in the direct space and in the reciprocal space, to really cover all the features of these glassy systems. So first of all, I would like to show you, for example, what you can learn when you investigate the systems with confocal microscopy. This is possible, for example, when you can label the components of the system with fluorescent dyes. And the very good thing of confocal microscopy is that you can image volumes of the system, and using particle tracking, you can obtain coordinates, and therefore from this you can calculate structural, so statistical functions which describe the structure and dynamics of the system, because you can also, of course, take time series of these volumes to follow the dynamics of the system. And so in this sense here, I'm showing you what we could obtain for a mixture which presents a large asymmetry in the components with the size ratio which is 1:5 between the small and the large particles at a high packing fraction. And as you see from confocal microscopy studies, we could determine the pair distribution functions of the large component, in this case in the mixture, as a function of increasing mixing ratio, so from the pure large particles to essentially almost only small particles, so to a small fraction of large particles. And at the same time, we could measure also the mean square displacement of the same system. And from the combined information of these two functions, we could reveal indeed the caging transition that is occurring in this system. So the fact that the large particles first form, of course, a glass which is characterized by caging by other large particles, but then this cage transforms to a cage of small particles at high fractions. And you see this is seen not only in the structure, but also, of course, in the size and the localization length that you can extract from the mean square displacements. Actually, sorry, let me come back 1 second to this slide. Actually, the nice thing of confocal microscopy is that you don't only get access to average quantities, but you can also study actually local properties, which for glasses, and as I will show you later, even for more heterogeneous systems like gels, are fundamental aspects of these states. So you can analyze, for example, neighbor distributions, formation of clusters, and also characterize the dynamics locally. For example, you can define dynamically active regions with respect to the average dynamics. And so I wanted to show you this example of the study of, in this case, an asymmetric mixture, which is even more asymmetric, which, with a ratio of 1:10, similar packing fraction, and investigating the neighbor distributions and the cluster size distributions. We were able to show that in this system, on top of glassy states, we also can observe formation of gel states. And for example, we could also characterize indeed the presence of heterogeneity in these gels by calculating also functions which essentially determine indeed heterogeneous parameter, like for example this remoteness, which is essentially the way of calculating the size of voids from the average distance of points in the void from the surrounding particles. And not only you can characterize local structures, one can characterize also, as I said, local dynamics, and therefore find results on dynamical heterogeneity of the system. Here for the same mixture, you see an example in which I was highlighting basically, in these snapshots, you see the 20% faster particles in the systems. These are indeed the systems which form attractive glasses and gels. And you see that these faster dynamics are connected with actually the average number of bonds that these particles have, which are the particles which are closer to voids and indeed are, of course, the ones presenting faster dynamics. Not only that, we can combine also, for example, information on the dynamics with information on the structure. Here we were able to define the arrested particles within the set of particles investigated in a sample, and then define clusters of dynamically arrested particles, and we were able to see how the distribution of these clusters of dynamically arrested particles are very different, actually, depending on the glass state that we are investigating. So this is what the confocal microscopy can give us. So both average information on structure and dynamics and local properties. However, it has also some limitations. For example, the resolution sometimes, especially for largely asymmetric mixtures, becomes a limitation factor in terms of both spatial resolution but also time resolution for the acquisition of the data. So it is very useful to have also measurements of rather scattering-like experiments or also correlation, indeed dynamic scattering experiments or correlation techniques. So of course, from a scattering experiment, one can investigate average structure with the structure factor. What I would like to show you is rather what one can get concerning the dynamics. First of all, because the advantage of scattering is that we can analyze dynamics which are dependent on the wave vector and therefore give us information on the dynamics at characteristic length scales. And we found that a very useful tool for investigating mixtures is differential dynamic microscopy, which has the advantage of being combined essentially with the use of, for example, fluorescence, which is, of course, the way of being selective with respect to the species that we are investigating. And so it's particularly suitable to separate the dynamics of the species. And in this technique, essentially by correlating in the Fourier space image differences acquired with the microscope, we can extract the intermediate scattering function, like in dynamic light scattering. And on top of the selectivity, indeed, we can also overcome problems of resolution of the microscope. And for instance, in this study, we used DDM to investigate the dynamics of small particles, again in asymmetric mixtures with different asymmetries. And in this case, the fact that we could determine the Q-dependent dynamics was very important to reveal the existence of anomalous dynamics at a certain mixing ratio with logarithmic decays, which was a very interesting finding of this work. So finally, to mention another important technique, we should consider also rheology, which is a more macroscopic technique. So it's not, of course, microscopic like the techniques that we just saw, but it's a technique which is very sensitive, again, to reveal, for example, transitions in the glass states that we observe. Here an example again from our mixtures with different size asymmetries, in which by investigating the linear moduli or the yield stresses and strains, we could clearly see again a transition between different glass states composed by mostly large and mostly small particles, and the melting essentially associated with this transition. And actually, we could also see that rheology is dominated in these systems by the species, indeed, which is essentially the one which is gauging the other particles. So let's say the gauge dominating species in some way. And this, for example, could be done by extracting the components of the rheological response by comparing experiments with simulations. And indeed, we could see that, for example, the large particles were dominating the average response at a small fraction of small particles. But then, of course, there is again a transition to a response dominated by the small particles. So now, I think the other important bit is that the combination of these different inputs from the experimental techniques can be used then to understand our findings based on non-equilibrium theories of the glass transition. Here again, an example in which we used the structural input coming from confocal microscopy and the rheological measurements that we just saw in the previous slide to essentially compare, for example, to findings of mode coupling theory in this case. So the structural findings were used as an input to guide the MCT calculations, and based on the structural input, the MCT results were able to reproduce to a good degree of agreement the experimentals and also simulation findings. And another very nice example from a recent work, I think Luis will tell more about this work, is the comparison of results obtained in experiments with DDM, with differential dynamic microscopy, and the theory, in this case, the theory developed in the group of Magdaleno. And this comparison between experiment theory and actually also molecular dynamic simulations, I think it's the first real sound confirmation of what happens when we study transitions between fluid, double glasses and single glasses in these asymmetric binary mixtures. So now this is, as I said, for the first part of the talk, and now I will try to be quick on the second part, which, as I said, is a bit preliminary. So the idea is that we can use a similar approach, of course, to investigate also the other side, basically, of these non-equilibrium arrested systems, which is gel systems, which are formed by attractive colloids. And so the most used, most investigated system of this kind is mixtures of hard spheres and non-absorbing polymers. And a lot of investigations have been done on this system, indicating the importance of arrested phase separation for explaining the gelation mechanism, at least up to certain colloidal volume fractions, and for example, very important contributions to the understanding of gelation in the attractive systems and the interplay with indeed phase separation and also glass formation were provided again by the group of Magdaleno in the last years. But actually our interest was now shifting as it was, I would say in general, the research in the last years to soft systems, so soft colloids, in particular the well-studied PNIPAM microgels, which due to the responsiveness, for example, with respect to temperature changes, are very used systems also in applications, for example, for realizing delivery systems or membranes which are thermoresponsive, but are also studied as a model system indeed of soft colloids. And so the question that some people recently tried to answer is how is depletion interaction essentially changing in these soft systems, so soft colloids. And so there are not actually many studies, but there are some recent studies from the group of Lorenzo Rigatti in collaboration with Peter Schurtenberger, which showed that while the initial expectation was that depletion was actually enhanced by the softness of the colloidal particle as an effect of the possible interpenetration, actually in the real systems, in mixtures of PNIPAM particles, this was not the case. Actually, depletion was found to be rather weak, and this was explained essentially with the fact that the density of the soft colloids is not uniform, so you have to consider that the core of the particles is harder than you would expect in a first approximation. And then there are also other effects which might affect the depletion, which is the possible penetration, for example, of the depletants inside the microgels. So what we were wondering on top of these effects is how these changes in depletion might influence the gelation of this kind of systems. So while this is a goal that we didn't achieve yet, so I hope I will show you that we found some interesting thing in the way to study that. So actually what we decided to study was not a mixture of microgels, but rather really a system closer to the classical art sphere polymer mixture, which is a mixture of PNIPAM microgel and polystyrene sulfonate, which is also a system for which there are some old preliminary data which indeed show the presence of aggregation with increasing the amount of polystyrene sulfonate at fixed concentration, let's say, of the microgels. So you see here, we have large fluorescently labeled microgels mixed with PSS at different concentrations. We have a low volume fraction of the microgels, and this is a short range estimated attraction in the system. And we investigate in the swollen state of the microgels at room temperature. So the first observation that we had is something interesting, which is the fact that these systems, when you had the PSS, indeed aggregate, but the kinetics of this aggregation is quite slow, which is interesting for the experiments because we can essentially monitor it with time-dependent measurements, which is typically very difficult to do with the art sphere polymer mixtures. And then, first of all, we looked at the steady states of the samples. So after, let's say, aggregation took place, and for different effective volume fractions of the polymers. And as you can see, we see the what we expected, and also what you could expect from colloid polymer mixture in general, which is that at some characteristic volume fraction of the PSS, we observe aggregation indeed in the structure, and also the slowdown of the dynamics, essentially, which become more and more arrested. So far, let's say something expected, and it can be seen also by looking at the evolution of the height of the peak of the pair distribution function, or the position of the peak, with a very clear transition, and also in the effective diffusion coefficient as a function of the PSS concentration. So we can distinguish clearly between sort of liquid states and aggregate gel states in the system. And now, as I said, in this system, we can investigate actually the kinetics of the aggregation. And for example, you can follow the evolution of the pair distribution function, of the mean square displacement, and the bond distribution with time, which evidently indicate the progressive aggregation. So we did that for all the systems that we investigated. And there is where we found something that I would say it's interesting, which we don't understand yet completely, but which is sort of unexpected, I would say. So, of course, for the liquids, here we monitor the average coordination number as a function of time, of waiting time. So for the liquids, there is essentially no evolution, while for the gels, for the aggregate states, we find that there is, of course, an evolution with an increasing number as a function of time, but the interesting finding is that this kinetics becomes slower and slower with increasing the PSS content. So if we quench the system more into the, let's say, gel state, the kinetics becomes lower, which is sort of counterintuitive, I would say, to some extent. So to get a bit more information on this kind of transition, we analyzed also our data in terms of DDM, and from the intermediate scattering functions, we extracted average relaxation times from fitting the data with stretched exponentials. And then we plotted basically the evolution with waiting time of this average relaxation times. And for systems which are essentially at the onset of aggregation gelation, we found a progressive evolution of this relaxation time. And if we look at the time dependence of this data for different Q values, they seem to be compatible, for example, with expectations of the LCA aggregation, so with the fractal dimension of the LCA, basically. But then, if we switch to the systems which are deeper in the gel state, the behavior is completely different. You see that we find a long latency period over which the dynamics do not really seem to evolve. After that, there is instead a very sudden slowdown, which a dependence which is much steeper than what is would be expected, as we said, for the LCA. So it's a very, very different behavior. which is seen also in the Q dependence of these relaxation times, which in the first case, it's still diffusive at the initial times, and then it evolves, but with no very clear transition to have a different Q dependence. But in the second case, for a deeper quench, you see that there is a sudden transition to another Q dependence, which is Q to the -3 in this case, which is indicative, as also shown by some recent works on polystyrene PNIPAM induced gelation. The induced temperature-induced gelation is characteristic of a network formation in this kind of system. So this is my question for you. So, as Magdaleno says, I think it's also the scope of this meeting to ask you if you have any ideas of why these kinetics might be so different, and so try to find an explanation for this phenomenon. And with this, I think I'm ready to conclude, so I hope I was able to show you that essentially if we're investigating this non-equilibrium glassy states or gel states also, the combination of different techniques is essential to cover all the aspects of all the properties of these systems. And I was showing you these recent results on the soft depletion mixtures, which show these anomalous kinetics, which we would like to understand. And thank you for your attention.
Thank you very much, Marco. Okay, we have time for a quick question so as to move on to the following talk. So there are several questions. I have myself some, but let me see if from the audience we can collect presentations. Marco, could you please
stop the sharing?
Thank you very much. Now, so for me to see if there are questions and to be able to. Okay, right now there are no questions in the chat. And how about here in the audience? Remember that you can push the button if you want to make a question. Put the button in the upper right side of your screen that says share the screen and the microphone, and go ahead with your questions. I'm referring to the audience. By the way, it was very unfortunate that we could not arrange, let's say there are several people who should be, let's say, in the screen right here, but they didn't push the button, so we cannot promote them on our own. But we people both in Mexico and abroad. So anyway, but for the time being, questions from the audience.
May I ask one question?
Please go ahead.
Yeah. So you had a transition line that was called a transient gel. So
Yes.
The transient gel, is that a percolating state that is different each time?
Yeah, so yeah, that's the distinction essentially that, well, this depends, of course, also on the packing fraction of the colloids, but essentially it's normally the percolation state that you find before eventually entering into, so crossing the glass line or the spinodal decomposition line, essentially, and which typically has some mechanical stability, basically.
And a true gel is then a percolated state that doesn't change over time anymore, right? So do these two lines meet somewhere, or?
Well, you see, I mean, there is, I think there is no clear, at least experimental indication, because it's very difficult typically for the experiment to really see where you enter for example, the spinodal, because it's preempted by the percolation, no? So essentially you stabilize the system before, but of course Magdaleno has done, I mean, the group of Magdaleno has now in the last years several papers where they show, well, they mostly show essentially where the glass and the spinodal line cross, so how they essentially you can find where you enter one or the other. Of course, the transient percolation, it's more what we observe in the experiments, let's say. So as far as I know, there is no very clear experimental theoretical work that shows where these lines are, basically.
But you can so determine the true gel under the so in the unstable region under the spinodal by doing a sequence experiment, right? So that is possible. But for the transient gel line, that will be more difficult, I guess.
Yeah, exactly. Yes. Yeah.
Okay. Thanks.
Okay, let me see. Leticia, you have, do you want to make a question or comment?
Yeah, no, a question. When you have this slope that is equal to 3, you move to very low Q or high Q?
No, actually, what I was showing is the tau against Q, no? So essentially, this slope of -3 is for the long times of waiting times, basically, of the systems where we do deep quench, basically.
Oh, yeah.
In the system, no? So the tau versus Q is showing this Q to the -3 dependence for a certain region of Qs, which is from the low Q, the lowest accessible Q, to some, let's say, intermediate Q values. And then there seems to be a Q where it becomes essentially almost constant, which is also what, for example, it was observed in this other work that I was mentioning, so that you can see 3 different regions, essentially, of the Q dependence of tau. So one which is essentially what we would see should be the large-scale region, where you see the whole gel, more or less. Then there is an intermediate where you see intermediate scale structures, and then at large Qs you should see like the fractal internal structure of the system. But in our case, we were able to access only sort of the low Q part of.
Okay, thank you.
Okay, let me see. Let me see here.
So one short question.
Yeah, please.
So if you have very short range attractive interactions, so like sticky spheres, and there are experiments where it is shown that the gel line crosses the critical point. Is that something that you also see in simulations that you or other people did?
So I don't know. I mean, I didn't do simulations in these systems.
Calculations.
And yes, I mean, no, I at least I cannot say that we observed clearly this transition in at least in our. I mean, in the experiments that I was showing. Today, we don't really have yet a description, a quantitative description that would allow us to compare, to find this line, basically. In hard sphere colloid mixtures, I think there are, as you say, already data which show that.
Okay.
Okay, we don't have more questions in the chats. and nobody else is raising his hand. So let's then thank Marco for his talk. We will still have time in the after Luis's final presentation to discuss. I myself have a question, not a question, but something to discuss with respect to gels, transient clusters, and so on. But we'll come back to that after Luis's presentation. So thank you, Marco.